Good morning, San Antonio. I'm Donna Rivera, Associate Director for Pharmacoepidemiology in the Oncology Center of Excellence at the U.S. Food and Drug Administration. Today, I'll be speaking with you about how COVID-19 changed evidence generation, an OCE perspective. Is on how COVID-19 has changed various aspects of cancer care, research, and drug development. COVID-19 has fundamentally changed a variety of things, including approaches to evidence generation. In this context, I think of COVID-19 as a catalyst. A catalyst can be defined as a substance that increases reaction rate by altering the reaction mechanism. Catalysts have definitive properties, including accelerating the rate of reaction, and in this way, COVID-19 required a rapid response. Catalysts are selective and specific, and it was required to develop public health strategies for healthcare treatment and risk mitigation, and required quality evidence for specific interventions. Catalysts also lower the activation energy, which is the energy barrier that must be surmounted for a chemical reaction to occur. Lowering that energy of activation reduces typical barriers. As a result of these actions, catalysts make it easier for atoms to break and form chemical bonds to produce new combinations and new substances. So how can we think about how those barriers were reduced, the differences in approaches during COVID-19, and what could be sustained as permanent changes or new combinations? Catalysts can also be enhanced by a supporter or promoter, and it's important to think about the system that is needed to affect or promote such changes. Finally, a catalyst can also be a person or thing that causes a change. And in areas where we can make advances that benefit patients, we should all strive to be that change. At the intersection of COVID-19 and cancer is a lot of complexity, and it includes various components, such as COVID-19 infection and post-acute sequelae, COVID-19 vaccination and immunity, including understanding of immunity and breakthrough infections in the immunocompromised populations, continuity of care and treatment, cancer prevention, the impact of COVID-19 on clinical trials, and public health communication. To approach the challenges associated with COVID-19 for patients with cancer, there have been a variety of efforts ongoing, and I'm going to focus on key areas that have impacted evidence generation, the first of which is regulatory flexibility and modernization of evidence. The FDA has issued multiple guidances related to COVID-19. Perhaps the most recognizable to this audience is the guidance highlighted here on the conduct of clinical trials. There are various other guidances available for reference. In this specific guidance on trial conduct during the COVID-19 pandemic, the development was based on the following guiding principles, of which the most important was safety. Ensuring the safety of trial participants is paramount. This was true before the pandemic and remains true now. The guidance document also provides information on the most common concerns, including those around patient protections, as well as alternative offsite methods and remote monitoring. It also describes contingency measures and the process impacts around COVID-19, which might include changes to trial conduct, protocol modifications or deviations, changes in data management and missing data, as well as COVID-19 therapies, including vaccines received under EUA. And of course, within the context of this guidance, contacting the FDA with specific questions is recommended. Of note, the FDA did not create new rules in this guidance and was already promoting many of these flexibilities pre-pandemic. While COVID-19 has changed many things and the pandemic is still ongoing, the OCE commitment to our mission remains unchanged. Here, I would like to highlight key approvals during 2020 and 2021 for the treatment of breast cancer, including agents for patients with HER2-positive metastatic breast cancer, the first antibody-drug conjugate targeting TROP2, and treatments for TNBC and hormone receptor-positive breast cancer. Additionally, during this time, both patients and practitioners may favor reducing healthcare system interactions to reduce the risk of COVID-19 transmission. Approvals highlighted here show an increased availability of medication administration options, including tucatinib as an oral administration, pembrolizumab with alternative dosing regimens, and also the subcutaneous administration option. Another focus during the pandemic is around broadening eligibility criteria. Listed are several guidances that have been released between 2020 and 2021 that are specifically focused on advancing this area. Of note, in August 2020, there were two guidances released for breast cancer, one on male breast cancer and the other on premenopausal women with breast cancer. Decentralization is another aspect that has accelerated during COVID-19. Decentralized trials have several potential benefits, which include reduced patient and sponsor burden and increased accrual and retention of a more diverse trial population. Since modifications were done in the middle of large ongoing cancer trials, and because industry is deploying aspects of these trials to respond to the COVID-19 pandemic, the Oncology Center of Excellence is interested in collecting data specifically on remote assessments and trial site assessments by sponsors by asking to voluntarily add flags in the data sets of NDA and BLA submissions. This could include information on items such as remote clinic visits and telemedicine, as well as remote administration. And this is an opportunity to evaluate the effect of remote assessments on trial data to advance decentralized trials in oncology. How can we take this opportunity to learn from change? COVID-19 has forced drug development to think in many new ways and catalyzed different areas of evidence generation, and that really has created opportunities. In the actions taken around rapid guidance issuance, trial modifications, and an emphasis on inclusivity of patient populations, this has led to adaptations around the use of decentralized approaches and opens the door to opportunities for the potential further use of real-world data. Based on the flexibilities needed and demonstrated use of real-world data in response, this might be an opportunity to focus on how we can accelerate methods and adequately utilize real-world data in a meaningful way. The second key area I'm briefly going to focus on is the role of real-world data in evidence generation. Let's begin with definitions from the FDA Real-World Evidence Framework. Real-world data are data relating to patient health status and/or the delivery of healthcare routinely collected from a variety of sources. Real-world evidence is the clinical evidence about the usage and potential benefits or risks of a medical product derived from analysis of real-world data. Real-world data sources include electronic health records, administrative claims data, registry data, and patient-generated health data, among others, that are comprised from a variety of clinical data types. The Reagan-Udall Friends of Cancer Research Evidence Accelerator is an example of the use of real-world data in public health response. Launched in April 2020, the COVID-19 Evidence Accelerator brings together leading healthcare experts across government and regulatory agencies, providers, insurers, and academic institutions in a collaborative effort to share insights and answer key questions to inform the collective COVID-19 response. Project Post-COVIDity was launched by the Oncology Center of Excellence when an early need was identified to elucidate understanding on the treatment of patients with cancer after recovery from COVID-19, when there was limited data available in this rapidly emerging situation. The aim is to characterize patients with cancer and evaluate longitudinal sequelae, how these potentially new conditions may affect treatment and safety of approved medical products, and questions around vaccination and immunity, by conducting a parallel analysis with data collaborators using a common protocol. Several data collaborations were developed quickly to evaluate COVID-19 related questions, including a collaboration with SciAps to characterize patients and a collaboration with Flatiron to characterize treatment, care, and patterns. Transitioning to innovative design approaches toward a learning healthcare system, presented is the evidence continuum with, on the far left, the randomized controlled trial, and on the far right, real-world data. Along this evidence generation continuum, there are several components of design and aspects presented. For example, in the randomized controlled trial, it is prospective, interventional, randomized with systematic assessment and evaluation frequency, typically using a highly monitored protocol, and has strict eligibility criteria. As we move further along the continuum, I'll highlight key aspects that are different. For example, in decentralized and hybrid trials, there is a difference in that they are highly monitored with the ability for remote assessment in potentially a virtual arm. Furthermore, in pragmatic designs, there may be pre-specified clinical practice assessments, such as the ability to embed, and there's selective monitoring. And finally, on the far right with real-world data, it is different in that it is often retrospective, non-interventional, non-randomized, with routine clinical practice assessments that may not be monitored. However, it does afford the benefit of the broadest eligibility of a population. COVID-19 has disrupted healthcare and forced flexibilities and considerations of alternative evidence generation strategies, including real-world data. What are the potential opportunities for the use of real-world data? There are opportunities in its use as complementary evidence, in understanding drug effects in underrepresented populations or molecular subgroups and rare cancers. Additionally, as well-designed external control arms in advancing innovative trial designs. There are, however, challenges, specifically challenges in the characterization and quality of real-world data, challenges to interpretation and use of real-world data in causal inference, and finally, in the use of real-world data for calculation and validation of response and time to event endpoint. I'm going to conclude by asking a few questions. What has changed during COVID-19? Cancer clinical trial conduct and evidence generation, as you've heard from my colleagues. Collaboration was present on an unprecedented global level. How did we adapt in oncology? By expanding access and increasing inclusion of specific populations. Where are there opportunities to increase modernization? The role of decentralized trials may play a fundamental role. How do we sustain what we've learned? In post-COVID, how do we continue to advance oncology drug development, build on the potential opportunities using real-world data that have been demonstrated, and build on those lessons learned to accelerate progress and share knowledge? The FDA Oncology Center of Excellence has focused efforts underway to advance real-world data, patient-generated data, digital health technology, and decentralized trials. Through all of these challenges faced, hopefully we can find the opportunity to create sustainable change and, most importantly, continue to advance patient-focused drug development. I would like to thank all of my wonderful colleagues. I'd like to thank the organizers for the invitation, and I'd like to thank all of you for listening. Please take care.